Thank you, Marta. My name is Alex, and I'm a phage scientist from Westmead Institute for Medical Research in Sydney, Australia. And I must say, I feel really honored and humbled to give this first talk at Phage Oxford 2021. And I thank the organizers for this opportunity. I'm also very lucky to have the opportunity to be involved in both basic and translational research when it comes to phage therapy. And today I will share with you the results of my latest research that involves the studying the L forms and their biology and their interaction with bacteriophages. So first of all, I will just talk a little bit about L forms. So what are the L forms? They are not any kind of isomers. They are defined by Emmy Kleineberger at Lister Institute. That's where the L comes from. And they are cell wall deficient bacteria or alternative bacterial life form. They partially or completely lack the cell wall, and they can be induced by different cell wall targeting antibiotics such as cephalosporins or penicillin, or immune effectors such as lysosine. And they are found in many bacterial species, well described in Listeria monocytogenes, Staph aureus, Bacillus subtilis, but it's still not clear whether this is a universal adaptive strategy in bacteria. They are common also in clinical samples, and here on this slide you can see examples of Mycobacterium tuberculosis cell forms isolated from blood samples. Unlike persisters, L forms continue to divide, and they are metabolically active bacteria, but they are less metabolically active than their cell wall counterparts. And on this graph, you can see this, the growth curves of cell wall bacteria and cell wall counterparts. And on this little movie from Jeff Errington and his paper in eLife, you can see the mode of division they are using to divide. So as we saw, they successfully divide, but they do not rely on this essential division machinery in bacteria, which is thought to be essential. Instead, the division of L forms depends on the rate of production of the membrane, of the cell membrane. So excessive production of the cell membrane leads to cell to change the shape, which then results to spontaneous separation into daughter cells. And here on this slide, you can see just some of the modes of proliferation that the L forms use to divide. And this is thought to be the ancient primordial mechanism of division used by bacterial ancestors. So last but not least, L forms have this unique ability to rebuild the peptidoglycan or the cell wall with withdrawal of antibiotic. So the aim of our study was to investigate how common is this L form switching in bacteria and in human pathogens. And for this, we used the model organism Escherichia coli. And we tested a total of 45 clinical isolates of Escherichia coli to see if they have potential to switch to L form state in the presence of last line antibiotic carbapenem called meropenem. Of these 45 strains, 2/3 or 77% had the ability to switch to L form state. And of these, 86 had ability to rebuild the cell wall within 24 hours of incubation after antibiotic withdrawal. Of these 45 strains, we selected 40 strains to do some further characterization and to check their susceptibility to meropenem in revertance to see if it changes after this L form switching, and we saw no changes. So they retain their sensitivity to meropenem after L form switching. Of these 14 strains, we selected 4 strains, and we did all genome sequencing on the parent strains, on cell wall bacteria and L form revertance to see if there are any changes generated during this cell wall to L form and then cell wall transition. And we saw no changes in 3 of them. They were identical to their parent strains. And in 1 strain, we saw some substantial some substantial changes at the genome level. As mentioned before, majority of tested E. coli form quickly reverted to their cell wall state just within 20 hours. And using advanced optical microscopy, we show that L forms can develop quickly, just within few hours. in semi-solid and osmoprotective L-form media. And that they can successfully proliferate also under aerobic condition, which is kind of unusual because other people reported that they are sensitive to aerobic conditions due to oxidative stress they are experiencing. We also tested the L-form sensitivity to other beta-lactam antibiotics and aminoglycosides and fluoroquinolones, and we showed that they are completely resistant to all beta-lactams tested, including cephalosporins and all antibiotics that are targeting specific penicillin-binding proteins, including PVP 2, 3, 4, 5, and 6. Unlike beta-lactam resistance, we show that L-forms retain their sensitivity to gentamicin and ciprofloxacin. We also wanted to understand better the L-form cell cycle and the growth dynamics. And again, by using the time-lapse microscopy, we measured duration of the L-form cycle in 20 selected consecutive cells. And we observed that first division occurs in most of them after 5 hours of addition of meropenem to exponentially growing bacteria. And those division events resembled previously described irregular shape perturbations leading to asymmetrical scission and generation of this heterogeneous population of meropenem-resistant L-forms. We measured that duration of the L-form cycle was on average 111 minutes with standard deviation, which was huge, around 77 minutes. We also tested survival rates of meropenem-induced L-forms under oxidative stress and show that they are significantly higher than in their faster growing cell wall bacteria. And this relatively high tolerance to oxidative stress may be a result from a priming effect of reactive oxygen species which are generated during this cell wall to L-form transition. We also analyzed the transcriptome of 4 selected L-forms, Escherichia coli, and showed that they do not suffer some extensive DNA damage. And you can see on these graphs on the right that LEX-A is slightly upregulated, and REC-A and low fidelity polymerase 4 DIN-B are slightly upregulated. And in case of one bacteria, you can see that REC-A is significantly upregulated, which again indicates the specific transcriptome profiles for some strains or genotypes. When it comes to oxidative stress response, OXCR, which is one of the main genes involved in oxidative stress response, is not upregulated, and LRB, which is the inner membrane protein responsible and essential in oxidative stress response in bacteria, is significantly upregulated in all tested bacteria except this one, which probably suggests this one is ST131 clade C, that this notorious pathogen is using some alternative pathways for in oxidative stress response. The next question we asked is what's happening in terms of phages, what's the interaction between L forms, which we think are clinically important bacterial forms, and the phages, and what is the implication in phage therapy. So this is largely an unexplored area. And why is this important? We think because phage therapy is advocated very often for recurrent and biofilm-associated infections where we expect to find the L forms. But there is a little experimental evidence to support that phage therapy is actually efficient in these settings. And we have different examples where T4 phage therapy failed in clinical trials where authors of the studies show that phages were effectively killing bacteria in vitro conditions, but were not successful in killing bacteria under in vivo conditions. I think my screen is frozen. Let me see. No, oh, okay, it's moving. So phage therapy is undeniably the third great invention after the vaccines and antibiotics, but we still have many questions to answer. We still don't understand the efficacy in humans and how they interact with phages with other phages or with antibiotics, and what are the implications of interaction with between phages and host immune system, and what's the best dose, and so on. And they are definitely highly promising antibacterials, but we know that sometimes they fail in patients. So obligatory lytic phages are the ones that are used in phage therapy, and they are thought to be ideal therapeutics because they have a huge potential to inhibit bacterial growth and biofilm, to reduce biofilm, and to inhibit formation of the biofilm. But to understand phage replication strategy, we need to also take into account the bacterial physiological state. Exponentially growing bacteria are shown on this little movie, and these bacteria are growing under optimal condition in the lab. And these conditions, based on exponentially phased bacteria, are based on all antibiotic and phage sensitivity testings. But what we think we can find in vivo conditions are stationary phase bacteria, so slow growing bacteria, persisters, which are not dividing at all, and then again, going back to these L forms, which are shown to be important in pathogenesis of many infectious. So those were the reasons why we decided to investigate interactions with therapeutic between L forms and therapeutic phages. And for that purpose, we choose two well-characterized myoviridae myoviruses. Two of them belong to T4-like phages, and ECO6 belongs to V5-like phages. And we used already developed L-form media in double layer format to test if these phages have the ability to lyse L-form bacteria. So again, we used the meropenem to induce the L-forms under osmoprotective conditions. And we saw that T4 phages, as you can see here, fail to lyse L-forms. And unlike them, V5-like phages retain their killing capacity in the L-forms. And this T4 failure, we again confirmed with growth curves. And this is just a screenshot of the plate with tetrazolium chloride, which is being used by growing bacteria and converted to the red formazan to track the metabolic activity. And we showed even with the growth curves that T4 phages fail to kill L forms. So we decided to determine the absorption rates in T4 phages to see if they are attaching to bacteria or not. And our results are suggesting that they are definitely attaching and interacting with the L forms as they do with their cell wall bacteria. So we decided to do pulse gel electrophoresis, and we show that upon infection of T4 phages on the gel, there is an obvious and distinct band which corresponds in size to ECO12 or T4 phages used in this study. And we also show that bacteria, when they revert, when they rebuild the cell wall after antibiotic cessation, they keep having this band in first few passages of bacteria, but then after some time, we think the bacteriophages are being segregated out. And for example, in passage number 5 and 10, you can see that the phages are absent. And in this particular case, we also used 2 restriction enzymes and show that actually this episomal phage is in a methylated state. And this is kind of logical because in phage annotation, we saw 2 or 3 methyltransferase genes. And we also identified few genes that are potentially involved in pseudolysogeny. And other people also showed before that stationary phase bacteria are actually supporting this pseudolysogenic state of T4-like phages, and that T4 are persisting as episomes during starvation, and that infection resumes actually when bacteria reaches some more appropriate conditions, and that this is all regulated by RPOS, which we showed previously is definitely upregulated in L forms. So just to conclude, L forms are slow-growing cell wall deficient bacteria, and they are definitely common, at least in E. coli, and may impair phage therapy efficacy in vivo. We show that T4-like phages are ineffective against L forms, but other less common phages such as V5, also myoviruses, retain their killing capacity in the L forms. That's why I hope I convinced you, L forms must be considered when we are deciding on phage or antibiotic therapy. And I just, again, just want to thank everyone involved in this study, and thank you for your attention, especially to people from UK and Newcastle, Kasia and Jeff, and to my supervisor, John, that made this all happen.